Okay, picking up where we left off, this is the second half of chapter 1. We talked about the scheduling and some of the aspects of pharmacology last time, and now let's dig into the drug development process. It's a laborious process. It's very, very expensive. It requires a long time to do, and for the very reasons that we talked about for the drug laws. You have to prove that it's efficacious, that it is safe, and does no harm. And so it is a tricky economic situation for pharmaceutical companies. They are often shown in a negative light on TV and in media, and maybe sometimes for good reason. But nevertheless, they have to invest significant amounts of time and effort in order just to bring a drug to market. And that brings its own challenges, not only in terms of the kinds of drugs that we see come to market, but also how the drug company is going to manage that drug once it does reach market. So in any case, we have a long time. And this is partly why, if you took my Bio 330 course, why I was so insistent on you doing journal review articles on model systems, because these are the seed crops of all drugs that enter the clinical system, with exceptionally rare exception. We see some type of an effect in a model system, and so we'll study a drug for 2 to 6 years. Oftentimes what will happen is drug companies will set up a model system of some kind, like cell growth or biologic growth or some other output. We call that an assay. And what drug companies have been doing over the last several years are trying to develop these high throughput assays where they can test their bank of compounds. We're talking about thousands or tens of thousands of compounds, and they look for hits on a given assay, like say biologic growth or cancer cell growth or something along those lines. And then what they'll do is they'll take those positive hits and start digging a little deeper to see if they can figure out why it's effective or what it's doing and that sort of a thing. And if they get to that point in roughly 2 to 6 years, a company then will sort of stake its claim with the FDA, saying they will file what's called an investigational new drug. And so that will allow them to begin the clinical process of evaluation. So the clinical process usually starts with a relatively small group, and this is teeny tiny when you figure that a drug that comes to market may see tens of millions or more patients. 25 to 50 is exceptionally small. In fact, it's probably limited to the smallest group they can get away with and still get statistically significant results. And that's usually a roughly 1 to 2 year process. What they do is they look at healthy volunteers, and what they're doing is establishing the safety profile. They are starting with extremely low doses and working their way up. It's a titration, if you will, looking for any toxic effects, looking to see how the drug distributes through the body, what we call pharmacokinetics. If, however, we're talking about an expedited, and that's going to be in the next slide, they may go directly to disease patients. This is particularly for either diseases that have no treatment or diseases that are terminal. If it gets through and they can establish a relatively good safety profile for healthy volunteers, they'll increase the size of the group. It may take 1 to 2 more years, and this is where they begin doing heavily rigorous research. They will do blinding, that is to say, the investigators may not know, or the patients will not know if they're getting a placebo versus a drug, or versus another agent if there's already an existing treatment out there. So these are patients that may or may not have the disorder, and they're looking, again, this is the initial trial for efficacy. If they can make it through this trial, and it's no guarantee that they will, Then they go into what's called the phase 3 trials, and this is usually a pretty big deal, and investors watch this one pretty closely. If something is cleared phase 2, that's a pretty good sign, or at least an initial sign, that it may get through phase 3 as well. It's still anyone's guess. And you're trying to shoot for over 5,000 patients in this case. These are double-blinded, where neither the clinician nor the patient knows what drug that they're getting, if possible. It's randomized. It's often crossover. That is to say, they'll have one group on the placebo and then one group on the drug, and then they'll flip them back. Multi-center, multi-country, if possible. And again, a 3 to 6 year period. Again, trying to get as many people as possible. Some diseases, that's not possible, or that sort of a thing. That's one of the barriers to drug development. And if they can get through and they have satisfactory results there, then they will receive FDA approval for a specific indication. There are times when clinicians will prescribe drugs that are not indicated for a given disorder. We call that off-label prescription. And clinicians are allowed to do that if they have full prescriptive authority. And it's usually reserved for MDs and DOs. They are allowed to prescribe off-label use with the caveat that they're at significant liability risk if for some reason something untoward happens. So you generally don't see a whole lot of off-label prescriptions unless the clinician is entirely confident that they can manage the situation from a legal perspective, if you know what I mean. But that doesn't even end. Even if we have FDA approval, that doesn't mean that the process is over. What we call clinical phase 4, this is the surveillance phase. This goes on basically forever. This is where you begin to see tens of thousands, hundreds of thousands, millions of patients on these drugs, and you're starting to get very, very strong statistical data if you're able to get it. What you're trying to do is get clinicians, you're trying to get patients to report any adverse effects so that they can begin developing probabilities or statistical analysis to indicate that there may be problems that were not seen even in these phase 2 and phase 3 trials. It's entirely possible. Several drugs have made it through FDA approval only to be yanked summarily due to unforeseen toxicities. Eli Lilly suffered from that with an analgesic back in the early 80s. And then not too long ago, Merck suffered a big loss with one of their analgesics that had cardiotoxicity. As I mentioned in the previous slide, there is an expedited process, and this came about because of that 1992 Congressional Act allowing for compassionate use and/or expedited approval. A nice review article about this expedited review process. It is fast, but it is also flawed. And the reason it's flawed is because you generally don't have the statistical power that you would have in a normal clinical trial. You don't have as many people, the studies are not quite as clearly defined, they may not be double-blinded, they may be the clinical trials may have drugs that are superimposed on existing therapies, lots of possibilities. So they're fast and they may help us to get to breakthroughs quicker. The problem is that they're flawed because they just aren't as careful, they aren't as carefully controlled, and lead to a risk of a sort of an allegorical assumption of efficacy, not unlike what we're seeing with hydroxychloroquine and COVID-19. So that process that I showed you in the previous slide takes many, many years and literally billions of dollars to see it through under traditional regulatory paradigms. And that leads to a pharmacoeconomic issue. And that is to say that many drug companies will not invest the capital and the trouble in order to get a drug approved if they don't have a market to bring that drug to. That is to say, any disease that has fewer than 200,000 patients could considerably be considered could be considerably classified as a rare disease. Even the fourth leading cause of cancer in the United States, the fourth leading cause, pancreatic cancer, pancreatic carcinoma, would be considered a rare disease because it only typically takes 35 to 40,000 patients in a year. So that's very much a rare disorder, even though it's the fourth leading cause of death. And so the Orphan Drug Act allows for funding of investigation of drugs. We call these things orphan drugs, because the pharmaceutical companies really do not have a fiduciary rationale for producing those drugs. They just can't afford to take a drug to market if it's not going to recoup the costs that way. Is one of the cold aspects of pharmacoeconomics. These two drugs right here were orphan drugs that I studied as a graduate student, peroxyl alcohol and farnesol, are both considered orphan drugs. The reason being, even though they might treat breast cancer, which is well over 200,000 patients, in those cases, these drugs are available from relatively straightforward sources. You don't get a patent. That's the other side of pharmacoeconomics, is that if you have a molecule and you have the patent for its synthesis, then you have sort of exclusive rights to it. This drug right here is a natural compound that is an estrogen inhibitor, and it's used in breast cancer that's estrogen receptor positive. Well, that can be isolated from natural sources, and so you just don't have a patent to make this, and so it's not economically viable for a drug company. So that's where orphan drugs come into. And orphan drugs fall also under the compassionate use clause that was written in 1992. That says that even though these drugs are investigational and may never get through the traditional clinical trials, nevertheless, they can be used for therapy even if they haven't been formally approved. The key is that they have to be in ongoing clinical trials. You just can't grab a drug off the shelf and go, oh, I want to try this in a cancer patient. There has to be some justification. There has to be some data trail indicating that there's potential efficacy. One of my hopes, and this is feeding off of the previous slide, one of my hopes coming out of this COVID-19 crisis is that we will have a restructured healthcare economic system. If you're a diehard market capitalist, that will sound like like anathema. But the truth is, we have so many different rare disorders out there, so many. The fact that we have to rely on profit making in order to get agents for these drugs created just doesn't seem morally correct. And it would be nice if we could find some other way to get that effort done to see an end, or at least a significant mitigation to all of these different rare disorders that are out there. In the meantime, if you find yourself in proximity to a patient, or if you're dealing with or are treating a patient that suffers from a rare disorder or an untreatable disorder, the Food and Drug Administration does maintain a website for drugs that fall under expanded access, that have been approved for expanded access, and or continuing clinical trials, investigational new drugs, list of approved and off-label uses for certain drugs. These types of resources are invaluable if you find yourself with a patient that is looking for additional options to treat their otherwise untreatable disorder. And so pharmacoeconomics are a barrier to drug development. It costs $1 billion, sometimes more, especially if trials have to be expanded or redone because of errors or insufficient statistical analysis. It's exceptionally risky from a financial standpoint. In fact, that may play into some of the, like the standard jokes about drug companies. What do you mean miracle drug? Well, it's made an enormous profits for us, so we call it a miracle drug in order to get people to buy more of it. That's the sort of jaded humor that's out there. But the financial risk and is significant. But the rewards are also significant as well. If you have a drug that you have a patent on a manufacturing process that no one else can take advantage of, it can mean exceptional profits for a company. Some other barriers, in addition to the pharmacoeconomic side, you may not have the ability to know if you have the disease or how serious the disease is. We're actually in the midst of that right now with coronavirus. We don't have the biomarkers to firmly establish or easily establish whether somebody has the COVID-19 or not. That's exactly what's going on right now. The tests that are out there have had issues. In fact, the CDC test didn't work for a period of time. The positive controls didn't work. So you didn't have a way of testing people. So lack of specific biomarkers. Another example of this was in Alzheimer's. In order to definitively diagnose someone with Alzheimer's, you pretty much had to have a brain biopsy, and usually that's not possible in somebody who's alive. So you could only diagnose them posthumously. Maybe we don't have an animal model that mimics the human disorder well enough. That's a common problem. That is to say, we would like an animal model that nearly perfectly represents the etiology and or the symptomology of a human disease, but most of the time we just have to rely on the best models that we have. My personal experience, we often would use rats for diabetes research, and unfortunately, rats can point us in the right direction, but not always give us the exact results that we might see in human trials. In fact, in some cases, the models would only work in male rats. We'd have to use male rats only. Couldn't use females because they wouldn't respond at all. Lack of consensus in terms of endpoints. If somebody has a disease, how do you define that disease? And that sounds sort of obtuse. Well, do they have the disease or don't they? Well, here's another example for you. This isn't as clear-cut as maybe I would like, but for instance, autism. A lot of the reason we saw a major jump in autism, or one of the reasons, I want to say this. First of all, folks having having kids later in life is a causality or a risk factor, but also in the early 2000s, if memory serves, the criteria for what defined autism and non-autism, or defined the autistic spectrum, shifted. And that ended up causing a whole bunch of patients that weren't previously listed under the autism spectrum then fell into autism. And so we saw this sort of jump occur. And that was more due to the change in the consensus, the guidelines for what defined autism, than it was the fact that all of a sudden there was this spike. All of these things, these first three things, are all barriers in what we would call the preclinical side of the equation. What are the markers that we're going to look for that allow us to determine if we have an effect or not? How are these models similar enough to human condition to be efficacious for giving us the right potential drugs for use in those disorders? And what are we looking for in terms of change in the patient or in the model systems in some cases that would define efficacy or define a clinical benefit, if you will? Some other barriers to drug development, and this is more in the phase 1, phase 2, phase 3. Sometimes we're reluctant, or we have been reluctant in the past, less so now, but it is still an issue that fewer women are tested in the clinical trials than men, and definitely low on the minorities as well. That we don't have enough statistical information about all of the minorities, not just talking about African Americans, but Asian Americans, Native American Indians, you name it. We do not have enough data, especially in the heterogeneous society that we live in here in the United States. It tends to be white, tends to be male. So that's one of the barriers to drug development, is that we don't have enough women, we don't have enough minorities, and that can lead to unanticipated side effects. Now, the Office of Women's Health was established in 1994 to counteract that. The NIH Revitalization Act injected some language in order to help encourage minority and women recruitment. And so it is having an effect, but we still have a ways to go. Lack of primary investigators, and this sort of a sore point for me, because as a preclinical investigator, I've heard a lot about this idea of what they call translational medicine. And I've had lots of friends who go on to medical school who have zero interest or minimal interest in participating in clinical studies. And I think that hurts the profession overall. That's another reason why, again, if you took my 330 course, you would see why I want to get you guys interested in research. First of all, it can be exceptionally lucrative, but the people that I know that do clinical and research feel, it's my impression that they feel like they have more rewarding careers. Their careers aren't as repetitive, they are challenged with new things every day, and they enjoy a fairly interesting professional lifestyle. I would strongly encourage you, if you can, to get involved in clinical trials and drug development. Even as an occupational therapist, there is plenty of room for people like you in that field. And so I want to encourage you to consider moving in that direction if possible. Inadequate collection infrastructure. Maybe we have patients, maybe we have investigators, but we don't have a way of collecting tissues or collecting biopsies, or the issue of managing patients in trials and so forth, and the complexity of the protocols, especially when you consider that if there's already an existing treatment, it is not morally feasible to deny somebody existing treatment for the sake of clinical investigation. Sometimes in the case of some of the expedited trials, if a patient has failed the first therapy, that is to say their disease does not respond to the frontline therapies, then they spill over into the investigational side of it, but they already have a tumor that's already refractory, and so that can lead to complexities in terms of investigation. Either superimposing the new drug on an existing treatment, or you're dealing with a refractory population to begin with that's already failed the frontline treatment. But all of these things right here on this side are involved in the clinical study, or shall we say the phase 4, if you will, the act of monitoring a drug once it's already been approved for a specific condition or treatment. So let's get into what a drug prescription looks like. You have a patient, or you yourself have been given a prescription. Let's look at the elements of that prescription, because it is possible that you might find yourself in possession of one of these, either as a family member or as a patient, maybe occasionally as a caregiver. So when we have a prescription, this is a sample drug prescription, you have to have the name of the person prescribing, the name and their official practice address and contact information. That's for tracking purposes. The higher the level the drug is on the schedule, the more tracking is necessary. And so this would be sort of a minimal tracking, if you will, the physician's name and their professional address and phone number. Have to have the patient's full name, patient's address for proper dispensary reasons. We don't want to be giving these drugs to the wrong person, as well as a date. And again, the higher the schedule number, in terms of going to Schedule 3 or Schedule 2, the more important that date becomes in terms of refills or in terms of length of the prescribed therapy. Then what we have is the RX side of it, receiveth thou. We call this the inscription. That is to say, the drug name, how strong the drug is, usually in units or in milligrams, and how many they are supposed to receive in terms of the number of pills or the number of units that they receive. Then below that, we have what's called the subscription. And this is pretty much everything down below in this part of the, maybe including the warning a little bit here. The subscription is basically instructions on how it's to be taken, how many refills, or the termination, the validity of the prescription. So we have the SIGNA, that's what SIG stands for, and these are the specific instructions to be taken with meals or twice a day, like BID, for instance. And sometimes these can be a little bit hard to decipher, but it's supposed to say how many you take and how often or under what circumstances you take it. Then we have the refill until, and this has become increasingly important with the abuse of certain types of drugs and the prevalence of pharmaceutical abuse. How many refills they're entitled to, or on what date does the prescription become invalid. And then lastly, they have to have a signature. This is a legal document, so the prescriber does have to sign in their own hand what the prescription is, and this also should include their DEA number and their state license number and so forth. The warning we don't see as much. Usually the pharmacies will fill that out. They'll often provide a sheet of warnings for potential side effects, do's and don'ts, and that sort of a thing. But there is a growing concern. As a care provider, you may not encounter this, but the fact that you may be serving as an advocate for a patient and so forth, this is a very, very real phenomenon, and it may lead to hesitancy or obstinacy on the part of certain caregivers because of their concern, both in terms of giving out too much drug, or in some cases, like in nursing homes and that sort of thing, they aren't giving enough drug. And it's just a little joke here. I'm not filling out this prescription because it's obviously forged and the doctor's writing is legible. But it is of a growing concern because it has been a trend for more and more pharmaceutical abuse, particularly in this age of opioid abuse. And what we find, if you look at the Drug Abuse Warning Network, if you look at drug-related emergency department visits, you can see that pharmaceuticals actually outpace alcohol or other illicit drugs. Okay, so we actually see many, many more pharmaceutical misuses, either self-medication or lack of compliance leads to issues with pharmaceuticals over alcohol or illegal drugs. Certainly females tend to be more than males. That's an interesting finding, that females tend to be more commonly showing up in emergency departments for pharmaceutical issues, and then overwhelmingly Caucasian. And I'm guessing that's probably partially an economic situation, but it's hard to say. So we see a lot of Caucasian Americans, relatively speaking, showing up, more females, and again, more pharmaceuticals than alcohol or other ones. A little bit about drug naming and classification, and this can be a barrier to treatment or a confounding issue as well, and that's why it's a good idea to cover it a little bit to help give you some awareness of that a drug can have several different names, whether it's its basic chemical name and so forth. And there is an effort to try to normalize these things. The U.S. Pharmacological Convention, USP, you might see that if you ever look in some of the literature on a drug or so forth, is designed to help create a standard frame of reference, because there are tens of thousands, if not hundreds of thousands, of compounds that are potentially medicinal in nature, even more than that probably, but in terms of what we already have. And so it can get a little confusing at times. So let's break out some of the ways that a drug can be named. And we're going to use this fun little drug down here on the lower right. Under the chemical convention, the American Chemical Society or the International Chemical Convention, we would call this N-methyl 3-phenyl 3,4-trifluoromethyl phenoxypropan 1-amine, which is certainly a mouthful. Okay, not going to sell a whole lot if you market a drug along those lines. Oftentimes we'll give it, the USP will give it a generic non-proprietary name, like for instance paroxetine or duloxetine. These are all the same. These are related to this one. This one just happens to be fluoxetine. Okay, it's a very popular antidepressant that we know of by its trade name Prozac. But for different purposes or for different indications or in different countries, we might call it Animexon or Serafem or Atifen or Deprex. It will depend on the formulary of the fluoxetine, what it's mixed with and what form it's in, what use it's intended for, or perhaps what country it's being approved for use in. For instance, we call it acetaminophen in the United States, but if you go to the UK, they call it paracetamol. And again, that's the non-proprietary name. So the chemical name is right here for this drug. Here is the generic name, and then here are some of the trade names, all for the same compound. In this class, I will generally default to the generic name if possible. Sometimes a name just takes hold and you just can't get it out of the lingo. Like for instance, aspirin. Okay, acetylsalicylic acid. Okay, we don't call it that. We call it aspirin, even though that's actually in fact a proprietary trade name. When it comes to those sort of non-proprietary names, we can even classify drugs not only by their names, but also by other schema as well. And you'll often see this in reference to modalities of treatments. So we will have different ways that we will call a drug, not just by its non-proprietary or even proprietary name. We want to call it by something that gives us a logical justification for why that drug's being used. For instance, what body system do we find ourselves working with? For instance, we might have a cardiovascular agent or a pulmonary agent or a neuroacting agent. So we're not really even calling it by its non-proprietary name. We're just saying these are the ones we might use for in this body system. The therapeutic action of that drug, for instance, is it an antihypertensive? Is it a hallucinogen? Is it a diuretic or something along those lines? It gives you a sort of, again, it explains a logical justification for why we would pick a drug. The mechanistic action, this is sort of closely related to a therapeutic, but it's usually a little more specific. A bronchodilator, vasoconstrictor, that sort of a thing. Molecular action, still even a little more detailed, like for instance, a calcium channel blocker could be a vasodilator. It could be something that we use for asthma, an anti-asthma agent that works in the lungs if I slide up in these things. Beta adrenergic agonist. Again, we're talking about a specific molecular action that achieves a mechanism, like for instance, a vasoconstrictor or a vasodilator or a cardiosuppressant, et cetera, et cetera. So these things are all interrelated, and I don't want to confuse you too much, but I do want you to understand that when we talk about some of these things in our class, we may be referring to molecular or mechanistic action, et cetera, et cetera. Sometimes the chemical makeup or the source. Beta-lactams are a classic version of this. Beta-lactams, those are antibiotics that act on a certain part of the bacteria. Plant alkaloids, we sometimes refer to it, tend to not use that very much in clinical settings. Usually talk about it when you're talking about the background or the history of a drug. When it comes to the non-proprietary names, the USP does like to assign names based on molecular or mechanistic function. For instance, a classic example of this are the drugs that inhibit an enzyme called the angiotensin converting enzyme. This is very, very important for blood pressure control. And in the case of these 10 members right here, they've all been given the last the last name Pril. So benazepril and moexapril and captopril and perindopril. They do that in order to help ease the introduction of a drug that may be unfamiliar to a clinician. And so by hearing the word Pril, they automatically begin to think, oh, ACE inhibitor. We also see this with OLOLs and OLs. Like OLOLs are beta blockers. They're non-specific. Whereas ALs may be a specific, what I mean by OL, may be a specific subset of beta receptors. And we'll talk about that a little later on. Lots of commonly prescribed drugs will have these sort of family names, if you will, in their non-proprietary lexicon. But this also can lead to confused drug names. Again, so many compounds, we begin running out of names for these things, whether you're talking about proprietary or non-proprietary. And here's just some examples of the drug name on the tan side with the drug name that it's often confused with. Guanafenicin and guanfacine, for instance. Guanfacine and guanafenicin. Two very, very different drugs. We don't want to be screwing up on those sorts of things. And so it does happen. The combination of the trade names, the non-proprietary names are similar enough that we want to exercise a little bit of care whenever we're in proximity to drugs that could be potentially confused. This slide completes Chapter 1. As always, if you have any questions or would like further discussion, feel free to reach me at any of these 3 locations. Thanks for your time, and I will talk to you again soon.